Welcome and thank you for joining today's Medline Canada sponsored presentation. I'm Shelley Masluk, Director of Clinical Engagement with Medline Canada, and I'll be your moderator today for this session. We'd like to thank Wounds Canada for this opportunity to educate in person as well as virtually. We also want to thank all of you for joining us today for this presentation. The Medline Canadian team is very excited to welcome Dr. Gregory Schultz, our guest speaker. In his presentation entitled Two New Technologies to Manage Chronic Wound Biofilms and Skin Folds, Dr. Schultz will share the science and data behind a slow-release iodine foam dressing and an antibacterial wicking sheet with hydrogen peroxide. Throughout his presentation, he will be asking important questions. Maybe he only knows the answers, but the answers will contribute to the improving your knowledge of both of these new technologies. During Dr. Schultz's presentation, I encourage our virtual audience to submit any questions to the Q&A box at the bottom of your screen. For our in-person audience, please use one of the floor mics to address your questions during our Q&A portion at the end of Dr. Schultz's presentation. Now, let me tell you a little bit about Dr. Gregory Schultz. He is a professor emeritus of obstetrics and gynecology at the University of Florida, where he served as the director of the Institute for Wound Research for 32 years. He graduated with BS and PhD degrees in biochemistry from Oklahoma State University, then completed postdoctoral research in cell biology at Yale University. A major focus of Dr. Schultz's more recent research is on understanding the role the bacterial biofilms play in stimulating chronic inflammation. He has co-authored over 420 scientific publications and been cited over 26,000 times. He's very smart, but he's also a very nice man. And if you'd like more details of Dr. Schultz's accomplishments, please refer to the Speakers Hub on the Wounds Canada app or refer to the session summary located in the virtual auditorium. Before I ask Dr. Schultz to come to the podium, we're going to have a poll question. So for anybody that's just come in, you can either scan or go into the website and get onto the Mentimeter. And here's our question. Which category of products do you primarily use for the treatment of biofilm? Is it wound irrigation solution, slow rinse iodine dressing, or silver dressings? Just to give us an idea of what you're using right now. Well, Dr. Schultz, what do you think of that? You see that? Silver dressings look to be the highest. We've got wound irrigation solution and then slow release iodine on the bottom. Okay, thank you. So it is my honor and pleasure to welcome Dr. Gregory Schultz to the podium.
Well, greetings, everyone. It's really a great pleasure to be back to Wounds Canada. COVID had a little bit of an impact on all of us getting together, but it's really enjoyable to be able to have face-to-face meetings again. So as Shelley has said, I'm going to take about the next 40 minutes, and I'm going to give you an overview and hopefully an update on two relatively new technologies that I think are very important tools in your toolbox to manage chronic wounds, especially the biofilm component on those chronic wounds, as well as a new product to help with skin folds and to control the bioburden that frequently provides much of the complications and pathologies associated with skin fold and skin fold management products. So as Shelley said, I am a consultant with Medline Industries and a board member of QuickMed Technologies, which is the company that developed the skin fold management product that I'll talk about. So let's sort of start and give you a little bit of a background of the history of iodine in wound care. And the reason we want to do that is because I want to review with you very briefly the importance of bacterial biofilms in chronic wounds. And you probably are aware now that in about the last 10 years, the whole field of chronic wound and chronic wound care has begun to understand much better the important role that bacterial biofilms play in, as I'll show you, creating an environment that is so toxic that the wound cells are unable to make progression out of the chronic inflammatory phase. And my wonderful colleague, Dr. Matt Malone, down in Australia, did a meta-analysis of published publications in this area and identified that within those publications, about 80% of the biopsies that are presented in those papers on chronic wounds contained bacterial biofilms, but only a tiny percent had any evidence of biofilms in acute healing wounds. Now, I'm going to drop into microbiology here for a minute. So remember those culture plates that you grew in your microbiology classes, and you saw these little tiny colonies that would grow up. That's the planktonic form of bacteria, rapidly proliferating, non-attached, can be very pathogenic forms of bacteria. But those same bacteria, in almost all cases, can convert from that non-attached, rapidly proliferating, antibiotic-sensitive form of bacteria into a different phenotype. And that's called the biofilm phenotype. Now, what causes that conversion into the biofilm phenotype is kind of complex, but basically, if the bugs get too high a concentration, they have a process called quorum sensing. It's sort of like hormones talking between bacteria. And when the bioburden of planktonic gets high enough, they will engage in the quorum sensing and trigger the bacteria to begin to express this different set of genes that causes them to attach, make this massive exopolymeric matrix that we'll look at that attaches them onto surrounding structures and helps to protect them from the predators that they experienced 1.2 billion years ago out in the environment, which are the amoebas that were the big single cell organisms that would engulf and eat bacteria. Unfortunately, that same conversion into the biofilm phenotype, as I'll show you, protects them for our amoebas, our neutrophils and macrophages that engulf and kill the bacteria. And also many, as I'll show you, the bacteria in the biofilm communities are not metabolically active. They're what microbiologists call persister cells. And the relationship and importance for us is understanding that antibiotics only effectively kill metabolically active bacteria. They do that by interfering with, binding, inhibiting, and activating the components of the bacteria, usually protein systems, RNA, DNA synthesis, transport proteins, etc., that are necessary for those bacteria to metabolize and proliferate. Well, if the bacteria is not metabolically active, those antibiotics have almost no effect. And in fact, laboratory studies show that bacteria that are sensitive to antibiotics in the planktonic phenotype, when they convert into the biofilm phenotype, develop over 10,000 times the tolerance to that same concentration of antibiotic. So basically, the antibiotics don't work well. Now, if the antibiotics don't work well and our neutrophils and macrophages, our immune system, can't engulf and kill these, how do we try to deal with these problems in the chronic wounds that you deal with? And that's what I'm going to talk about in terms of one of the products, the Ioplex, that can effectively help you manage that. Now, again, not to give you a microbiology headache, but this middle panel here, B, is a scanning electron micrograph of the surface of a chronic wound biopsy that Dr. Malone punched in one of his patients. And what you see is this massive amount of this matrix, this exopolymeric matrix that the bacteria have made to help attach and protect themselves in this form. Now, it also includes a significant amount of free extracellular bacterial DNA. About 20% of these components in most of these biofilm communities are DNA. about 80% is the polysaccharides that they synthesize and secrete only in the biofilm phenotype. But then we also have this RNA and DNA from bacteria. Now, the reason that's important for us to appreciate is that one of our evolved responses to try to counter this bacteria when they convert into the biofilm phenotype is our innate immune system. You remember we are all born with certain genetic abilities to combat bioburden infections. Well, our toll-like receptor 9 is actually evolved to recognize unique sequences that are only found in bacterial DNA. And this gets into the real microbial grasp, but these are called the CPG islands, and they'll find those sequences only within the bacterial DNA. When our toll-like receptor 9 expressing immune cells sense that, they immediately activate and begin to secrete more and more pro-inflammatory cytokines that draw in more and more neutrophils to try to help us remove and prevent biofilm reformation. In addition, biofilms are present on the surface of the wound, as you can see in the scanning electron micrograph, but also biofilms are present under the surface of almost all wound beds. And my wonderful colleague, Dr. Thomas Bjornholt at Copenhagen University, has quantitated this. And bacteria like Staph aureus can penetrate 50 to 70 microns under the surface of the wound bed. And that happens when the planktonic bacteria invade into the underlying dermis matrix of these wounds. They begin to quorum sense and they'll create biofilms there. Pseudomonas aeruginosa, which is even more invasive, can penetrate deeper and can go 100 to 150 microns into the base of the wound bed. Now, why is that important? Well, we need something that can penetrate through this upper layer of the wound bed and not get chemically inactivated, but can actually penetrate through to where it can begin to attack these biofilm structures that are under the surface of the wound bed. Also, in the International Wound Infection Institute revision that just came out last year, 2022, we talk about the slough project. And one of the keys is for you to appreciate that when you see these types of bioburden problems that's causing the chronic inflammation that leads to elevated proteases and reactive oxygen that destroy the proteins for healing, that's why the wounds don't heal, is that slough that's up there needs to be removed. But it's necessary, but it's not sufficient, because wiping the surface of a wound bed with something like a gauze or other superficial cleaning agent, yeah, that helps to get rid of the bugs on the surface, but it does not, in almost any cases, address this bioburden, the biofilm that's under the surface of the wound bed. And that's why the new international guidelines for identification and treatment of infections and wounds emphasizes surface cleansing, but deeper wound bed debridement to try to remove the bioburden, especially the biofilm that's underneath that. So how do we try to accomplish all of this? Well, in combination with good surface wound cleansing, one of the components that can help penetrate into the biofilm exopolymeric matrix, as well as penetrate to a certain distance within the wound bed, is to use a active iodine releasing dressing. And we'll talk about the Ioplex. Now, I want to make a big distinction. This is not a solution of iodine like betadine, because when we talk about the cytotoxicity of a burst release iodine product like betadine, it will, when you put it on a wound, will reach high concentrations that greatly exceed the cytotoxicity level for that particular type of iodine. However, when we use something like the Ioplex matrix, which is a foam that contains the iodine complexed within this polymer, it doesn't have this massive burst release. It has a controlled release. So our colleagues over in pharmacology talk about burst release and sustained controlled release. And the reason that's important are twofold. First of all, it doesn't typically exceed this cytotoxicity index. So it's not only, as I'll show you, killing the planktonic and biofilm bacteria, it's also not killing our wound cells. And because it is providing a sustained release, if the wound bed becomes re-inoculated with bacteria, such as ones that are now we know frequently around the edges of the wounds, or that penetrate directly through a membrane permeable dressing, that they're going to recolonize, and data that we've published with my wonderful colleague Randy Walcott down at Texas Tech and Lubbock, it actually will reform a tolerant biofilm in those patients within 3 days. So we don't want something that just kills like this. We want something and has high cytotoxicity. We want something that maintains this anti-planktonic, anti-biofilm treatment over a period of days, but doesn't exceed this cytotoxicity level. Now, again, this is kind of the chemistry, so I'll go through it quickly, but we heard today about the problems of using antibiotics under conditions that could potentially cause development of antibiotic-resistant bacteria. Now, remember, there's permanent genetic antibiotic resistant, like MRSA or VRE, where they have genes that permanently make them unable to be killed by the antibiotic exposure. That's totally different than the temporary tolerance that bacteria have when they are in the biofilm phenotype, because if we disperse those bacteria, re-expose them to the antibiotics, it'll kill them. But one of the nice things about the use of iodine is, unlike antibiotics that have a single target within a bacterial protein or DNA RNA system, iodine is a general oxidizing agent, and it will oxidize and iodinate multiple components within the bacteria. So it can both react with iodinate RNA DNA, soluble proteins, double bonds within the phospholipid backbone. And so it's almost impossible for a bacterium to develop genetic tolerance, that is permanent resistance to something like iodine that has multiple types of targets that will kill the bug, because the bugs can't evolve simultaneously resistant to all of these different targets. Now, there's a robust portfolio of published literature that supports the use of iodine's superiority in addressing killing bacterial biofilms. And I won't go through all of these again. These are essentially free downloads, but I would emphasize to you this new update from the International Wound Infection Institute's 2022 update. And one of the points that's emphasized in those papers, as well as in the IWI update, is that iodine releasing dressings are one of the most effective topical local treatments to kill biofilms. So if we grow bacteria into mature biofilms on the most relevant laboratory model we can, which is to grow them on pig dermal explants. Remember, the bacteria in your wounds are forming on dermal explants, on the dermis, like in the explants. They're not forming on polystyrene wells. Okay, that's another biofilm assay, but it's not very relevant for us clinically. And if we grow mature Pseudomonas aeruginosa biofilms for 4 days and then wipe off the surface and expose them to these multiple different wound dressings that contain or do not contain what the FDA calls preservatives. Okay, these are really antimicrobials, but the FDA, with its bizarre terminology, call them preservatives because they only allow you to talk about the dressing preservatives killing bacteria in the dressing or preventing bacteria from penetrating through. If you say this dressing kills bacteria in the wound, that's a drug claim. That's a no-no. So you have to appreciate a little bit of this semantic background of the FDA's relatively illogical way of how they try to regulate these dressings. But anyway, if we take dressings that do not have any preservatives, they will actually increase the amount of the bacteria that is present in this pigskin explant model. If we look at multiple other ones that contain different preservatives that will diffuse out of the dressing into the wound bed, and we assess how effectively will these reduce the mature biofilm bacteria, we can see that most of them don't really do a very good job at all. The only one that has incredibly effectiveness at removing, killing, and reducing the viable biofilm bacteria, that is log reduction, is the sustained release, slow release iodine. That's that green bar. Everything else basically doesn't have very much efficacy in this laboratory model. So this is where the Ioplex is able to provide this type of sustained release. So what is it composed of? Well, it's a polyvinyl alcohol, a PVA, so that's the material engineering aspect. It can absorb wound exudate, and at the same time, when it absorbs it, it causes a controlled release of the contained iodine because it is complexed with the polymers in the foam. And again, because it is sustained over a period of time, it's not burst release, it has very low cytotoxicity compared to other types of burst release antimicrobial solutions. It is normally black when it is in the fully charged state, has the iodine complexed in it. But as it absorbs moisture and begins to release the iodine into the wound, it begins to turn back to a pale or whitish color. And this is important because it helps you as a clinician to understand when the dressing is still being effective as an antimicrobial topical treatment. You have a very easy visual indicator of when it's still good or, oh, we need to change this dressing because the iodine has essentially been released. It's also, again, for the U.S., it's reimbursable by Medicaid and Medicare. And it also is convenient and it is non-staining. It doesn't cause a discoloration of the wound bed or the surrounding skin, so that allows you to have better visual estimation. Now, when we look at this, the slow release iodine foam dressing eliminates both Staph aureus and Pseudomonas in vitro. Again, this is using what's called the CDC bioreactor. That's where you grow the biofilm on a stainless plate. But I've also shown you where it can do it with Pseudomonas in the pigskin explant model. And when we look at the effectiveness on the two different bacteria, you can see that it is extremely effective compared to many of the other dressings that wound dressings that have different types of antimicrobial materials. And equally important, because now when we look at more recent data that uses DNA analysis of the bacteria in wounds, not just what you can culture, turns out that almost every chronic wound has a substantial contribution from strict anaerobic bacteria. Now, the question is, where are those anaerobic bacteria? They're usually in association with the biofilm, because the biofilm and planktonic bacteria are eating up all the oxygen, and they create a niche adjacent to the biofilm structures where there's very low oxygen. And if we actually put oxygen electrodes into biofilm structures in the lab, we can show the top of the biofilm where there are some metabolically active bacteria, but they're eating up all the oxygen. If we push that probe down to the bottom of the biofilm, there's essentially no oxygen getting down there. And that's where the anaerobic bacteria find an advantage. But the good news is that the iodine released from the Ioplex will also effectively kill the anaerobic bacteria. Again, most of the antibiotics that you would typically see used either systemically or topically don't work well against anaerobes. So This just shows visually, to help you get an appreciation for the effect, using confocal laser microscopy of a mature Pseudomonas aeruginosa biofilm. And what we can see is that when this is the control, in other words, it's not releasing iodine, that the green cells are viable. And if we look at the ioplex now in the same experiment, you see all of these red and yellow cells. Those are stained with a dye that picks up only dead cells. And so that shows the enormous effectiveness of the slow release iodine in killing these mature biofilms. Now, other products can have a little bit of an effect about killing these, but compared again to the ioplex and the sustained release form of iodine, it is by far the most effective treatment that you can use without causing high cytotoxicity. And that's just a 30-minute exposure. If we look at 24 hours, which is more relevant to what you would see in a clinical use, you can see the massive kill that the ioplex provides for these using the sustained release iodine. So, as I said, do all my antimicrobial dressings effectively kill biofilm on explants? And the answer is no. And again, when we use our pigskin explant model and we look at this, here again, this is the control. These are the non-preservative containing dressings. These are just like alginates that just absorb. They will actually increase the amount of biofilm bacteria. These antimicrobial containing preserved dressings will reduce about 1 log in 24 hours. But here is the sustained release iodine, again, within 24 hours in this relevant model, totally eliminates the bacteria in the biofilm. Now, this led to the most recent international consensus guidelines on identifying and treating biofilm. And this group, this consensus guideline that was just published, evolved what we hope is a clinician-friendly interpretation of all of that lab data and all of the other technical stuff that we talked about in what's called the step down then step up strategy. And what this tries to convey to you is that when you have a wound that's not been healing for 4 or 6 weeks, they come into your lab, they've got slough, they may have some subtle signs or maybe even a little more overt signs of local infection in the wound. There's almost 100% chance there's going to be a bioburden problem, both planktonic and biofilm. And because the bacteria in biofilms are almost totally tolerant to antibiotics and many of the antiseptic preservatives, the first key aspect about the treatment of wound bed preparation with biofilm-based wound care is to undergo aggressive debridement that is appropriate for the patient. Now, optimally, that might be a sharp debridement with a scalpel or a curette, but as you well know, many of those patients aren't necessarily good candidates for that. So you've got to come up with whatever is the most optimal way to aggressively debride the superficial slough, the bacteria on the surface, but also underneath that wound bed. Now, in addition, you need to combine that with a wound dressing that has the capacity to kill residual microcolonies of biofilm, because we can't always debride it out. There's always a little bit of biofilm microcolonies left, as well as prevent planktonic bacteria recolonizing the wound, because as I said, we've shown in clinical studies that if you use wet to dry gauze dressing after debridement, in 3 days, the biofilm is back in that patient's wound at the levels before you did the debridement. Now, when you do this type of good aggressive and frequent debridement, good antimicrobial and biofilm dressings, the chronic inflammation in the wound will begin to decrease, and you can go to less frequent, more maintenance type of debridement. You can shift to other types of bacterial biofilm and bioburden prohibiting dressings, and the wounds in most cases will start to heal with just that standard of care. But if you have patients that have other comorbidities, what you need to do is understand that they may need some extra help in beginning to heal their wounds effectively. And that's when you can step up to these advanced therapies like the combination cell and tissue products, growth factor, skin grafts, negative pressure wound therapy. And those advanced treatments will work very well because you have prepared the wound bed appropriately and gotten rid of the planktonic and biofilm bioburden that was causing the chronic inflammation that impaired healing. So this is the concept of the step down, step up. Don't start with your cheapest, weakest way to try to remove the bioburden because that's not going to do it. Get aggressive and use your most effective treatments first, and then step down as the wound comes out of the chronic inflammatory phase, then step up to advanced treatments as needed. So I'm going to shift gears now, and I'm going to talk about a new product that is available that is using our own natural reactive oxygen molecule to control both planktonic and biofilm-based bacteria that can form in the skin fold management products. And just to remind you that hydrogen peroxide, H2O2, is actually one of the two reactive oxygen molecules that our own body makes to kill bacteria. The other being hypochlorous acid, HOCl, which is generated from the hydrogen peroxide through the action of myeloperoxidases. So basically what happens is when the hydrogen peroxide that our neutrophils and macrophages make encounter a target, they will oxidize those. And they do that by oxidizing multiple targets, again, within a bacteria. So again, this is not like an antibiotic that has one highly selected target, which allows the bugs to make genetic permanent resistance. The hydrogen peroxide will oxidize multiple side chains of amino acids like the cysteine, oxidize that up to sulfenic acid, as well as create new disulfide bonds. It will oxidize the primary amines of arginine to make hydroxylamines. It will oxidize, add hydroxyl groups onto the double bonds of the lipid bilayer molecules. And when these molecules become oxidized, they denature those components, and that kills the bacteria. So the hydrogen peroxide is much like the iodine molecule that is also an oxidizing agent. It doesn't have one target in a bacteria. It has multiple targets. And this just shows where it can react with. So it can react with the glycoprotein side chains, the carbohydrates. It can oxidize the lipids within the bilayer, as well as the multiple disulfide bonds or multiple double bonds within the lipids. So how does our body generate hydrogen peroxide? You remember I told you this is one of the key reactive oxygen molecules. Well, it turns out when a neutrophil engulfs or a macrophage engulfs a bacteria, like shown here on the left-hand side, within those endocytic vesicles, also called phagosomes, that this enzyme complex called the NADPH oxidase, blah, 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 biochemistry complex assembles in the membrane. And what it does is it adds an electron onto oxygen, O2. That creates the superoxide radical O2 dot. Now, that is an incredibly powerful oxidizing agent. Unfortunately, it's so chemically reactive that it only has about a 30-second half-life within the endocytic vesicle. So it doesn't give it enough time to really encounter and kill all the bugs. So what our clever neutrophils and macrophages do is they secrete protons through a proton channel into that vesicle that converts the superoxide radical into H2O2, the hydrogen peroxide. In addition, it will generate the hypochlorous acid by secreting and putting into the endosome the myeloperoxidase enzyme that will add chlorines to it. Now, this happens incredibly rapidly. Within a matter of minutes, when a neutrophil engulfs a bacterium or fungus, what it will do is generate these multiple reactive oxygen molecules as shown by that false color thing. Okay, so the body generates this, but how does it limit the damage to our own wound cells during the production of the hydrogen peroxide? Well, we have evolved, fortunately bacteria have not evolved, the ability to neutralize this hydrogen peroxide to prevent it from doing off-target damage to our wound cells. And one of the most important molecules is a reducing agent like glutathione that chemically reacts with that oxidizing activity of hydrogen peroxide and basically generates oxygen in water again. And this just shows that when glutathione encounters a hydrogen peroxide molecule or the enzyme catalase, it will basically neutralize the hydrogen peroxide by internally oxidizing groups within the glutathione molecule. So glutathione is a sacrificial anti-oxidizing molecule that only we make to help protect us from our own reactive oxygen molecules. So how does this compare with other antimicrobial chemicals and developing ability to kill microbes? Well, as I'll show you, it is extremely effective in killing even the most difficult to kill microbes, including gram-positive, gram-negative bacteria, fungi, mycobacterium, and even envelope viruses. And just in a very conceptual way, when we look at the ability of different components to kill these different microorganisms, being spores being the very highest down, difficult to kill from enveloped viruses, it turns out that hydrogen peroxide, our own antimicrobial molecule, is incredibly effective at killing even some of the most difficult microbacterium. This is a visual picture that shows the typical lab results using FDA-required International Standard Organization tests, ISO tests, to make a claim of an antimicrobial or antibacterial material. And you can see when this new technology that sequesters the hydrogen peroxide in a dry form as a complex with zinc peroxide complex, that it will completely eliminate bacteria using these standard ISO tests. And when we quantitate that with methicillin-resistant staph epi, E. coli, Acinetobacter baumannii, you get at least 5 log kills, which is the threshold that the FDA requires to say this is an antibacterial preservative. Well, what's the clinical safety of this treatment? Because we want to kill the bugs and we don't want to kill our wound cells. It turns out that this sequestered hydrogen peroxide passed all standard FDA tests for cytotoxicity and skin irritation, meaning it is non-irritating, non-sensitive to intact skin. And that's in large part because the keratinized epidermal layer, superficial layer of the stratum corneum, is very impermeable to hydrogen peroxide, so it doesn't penetrate through intact skin to cause inflammation. The other aspect is that when we look at the concentrations of peroxide in different things, if you ever get your hair bleached, you're looking at 10% hydrogen peroxide when you go to the hair salon. If you look at a commercial cleansing agent, it's anywhere from 6% to over-the-counter disinfectants that contain hydrogen peroxide that you buy at Walmart. That's about 3%. The Drygo treated fabric is 10 times less than even the over-the-counter hydrogen peroxide you buy at Walmart. It's 0.3%. Now, the compatibility testing for Drygo, again, indicated that it passes all of the FDA for acute systemic infection, irritation, maximization, Klingman, and cytotoxicity. So blah, blah. This is a safe and effective material to use. And then what impact does it have on moisture management? Well, we want to have a fabric that draws the fluid, particularly the sweat that's accumulating. And you can see, again, this passes all the FDA standards for hydration and wicking. So in summary, what I hope I've shown you is that hydrogen peroxide is a major reactive species. It's made by our own neutrophils and macrophages. It kills the engulfed bacteria. But that helps to reduce the infection that can occur when these bacterial and fungal species colonize the wound. We have our own protection systems of catalase and glutathione that only we make, that the bugs don't make, so we don't get our own cells damaged compared to killing the bacteria. It uses the only advanced technique that can produce fabrics that contain hydrogen peroxide that's sequestered in a stable, dry state. So when it gets hydrated, then it releases the hydrogen peroxide. This is the only technology that's ever been developed and patented to do that. And fabrics that contain this immobilized, releasable hydrogen peroxide in the stable complex have powerful antibacterial and absorbent capacities. And I didn't emphasize it here, but it also is very effective against fungi and yeast. So these properties provide an excellent clinical management of skin folds and other areas of skin contact that help to maintain and regulate moisture imbalance, as well as providing effective antibacterial and antifungal effects. So at this point, we're going to stop and go for questions and answers.
Yeah, so thank you, Dr. Schultz, first of all, for sharing your experience with the science of the iodine and hydrogen peroxide and how they can clinically manage chronic wound biofilms and skin folds. Before we go to the questions, can we bring up one more poll question? So now that you've heard the science behind the effectiveness of hydrogen peroxide, would you consider using Drygo HP for your skin fold management? So of course, we're hoping that yes is big.
I obviously failed with 2 people, so I have to figure out what issues that those people want to know about.
Okay, so now's your opportunity. If anybody has any questions in the room, please go ahead.
Hi, I'm a registered practical nurse. I do basic, advanced, and diabetic foot care. I was told before that when I do a callus reduction, sometimes I come out cross wounds, I'm to use sterile water and then iodine, and then send them over to wound care. Is it better to use hydrogen peroxide and then use iodine? Like, are you going to knock each other out, or can they be used at the same time?
That's a good question. And let me go back a little bit. You know, it depends on the concentration of the hydrogen peroxide, because as I showed you, if you go to Walmart and you buy the over-the-counter bottle of hydrogen peroxide, that's 3%. You pour that into a wound, it's going to kill a lot of bacteria. Unfortunately, it's also going to kill a lot of wound cells. So it has to do with the concentration and the duration of exposure to the hydrogen peroxide. Part of the wound bed preparation biofilm-based wound care is to do effective cleansing. And as my wonderful colleague, Dot Weir says, don't anoint the wound, but cleanse the wound, right? So you need to have some aggressiveness in terms of your cleansing. That's both physical cleansing, but also using an effective antimicrobial. Because I won't go into all the data, but there's lots of data that shows that if you cleanse a wound with sterile saline, you get rid of the slough, but you don't do diddle to the bacterial bioburden. So that's why it's important to combine the wound cleansing with an effective antimicrobial that can kill planktonics within a couple of minutes exposure. Probably won't do very much on the biofilm because we have to do the more sustained release form of that. But it's better than just wiping it with sterile saline. And then combine that with effective debridement appropriate for that patient, and one of these dressings like Ioplex that will provide sustained anti-planktonic, anti-biofilm treatment. And that's in part of, if you look at that new IWII 2022 guidelines, it goes into those concepts and explains it a little bit better. But you know, it's always a balance that's called the therapeutic index. How effectively does this treatment, solution, dressing kill bacteria but doesn't kill wound cells? And our colleagues in pharmacology use this term called therapeutic index. It's the ratio of that. You want something that has to have a real high concentration before it kills our wound cells. That's the numerator. The denominator is how much of the product begins to cause cell death. And we want that to be a very, very, I said it backwards, very, very high number, and the antimicrobial effect at a very low number. So bottom line is, it's a balance to try to come up with the right therapeutic index of killing the bacteria, but not killing wound cells.
Hi, just have a question about the concentration. Does it create any kind of effervescence? And then, of course, there would be the complication of an embolism occurring if it did, right?
Right. So the Drygo product, when you anoint it, doesn't make the bubbles that you typically see if you pour some hydrogen peroxide solution in. And part of the reason is it provides a relatively sustained release of the hydrogen peroxide, unlike when you just pour hydrogen peroxide in, it breaks down, produces lots of oxygen and hydrogen and other stuff. So no, you're not going to see it bubble, but
Okay.
That's good in one sense, because that provides that sustained release, not this burst cytotoxic release.
Okay, thank you.
We've got some really
Over
interesting.
here.
It's just like a follow-up for what he just mentioned, because previously, like in my olden knowledge, only normal saline is the best solution for cleaning wounds. We were told before on some of the conference that I attended that never use hydrogen peroxide for the reason of what he had said, the effervescence, and it will go in air embolism. So is it safe to say now that the Drygo is basically only used for superficial wounds and not deeper wounds?
So your question is really appropriate, because the typical concept of using hydrogen peroxide in wound care is to pour that 3% hydrogen peroxide into the wound bed. Please don't do that. It has too high a cytotoxicity, and it is not providing a sustained release to allow the hydrogen peroxide to penetrate and kill the biofilm. We need some time for that hydrogen peroxide to penetrate into that matrix. So this in no way is meant to imply that solutions of hydrogen peroxide that you would typically be able to obtain are safe and effective in wounds. So the answer is hydrogen peroxide at the right concentration in the right delivery format is really effective, but don't pour hydrogen peroxide in wounds.
Hi, I'm a Swan nurse, and I wanted to know how you feel about using products like Vash cleanser or Prontazan as a wound bed preparation and a compress prior to using the Ioplex. How do you feel that that would benefit or hinder?
Okay, and that's a great question, and I'll answer it by saying there is no one golden pathway to treat every freaking patient. You have to adapt it to each patient. And the good news is that you can combine multiple types of treatments of the wound bed to take advantage of each of those different products' ability to do certain things. So for example, you talked about the Bosch. It's very effective. I work with Ergo. It's very effective, especially when it's used in negative pressure installation. It is phenomenally effective. We worked with OKCI about this, and it is incredibly effective at removing and killing bacterial biofilms in orthopedic implants and really chronic wounds. So there is definitely an application for hypochlorous acid, not sodium hypochlorite Dakins, that's too cytotoxic, but that's used in a kind of a different clinical application. Now, the Prontazan, blah, 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 here gets into the chemical structures that uses the preservative of polyhexamethylene biguanide, PHMB. It's a long cationic surfactant with positive charges. That doesn't kill the biofilm as effectively as hydrogen peroxide, both in these relevant clinical laboratory models as well as in initial clinical studies. Doesn't mean that Prontazan can't be used effectively in an initial cleansing. Yeah, but it is not as effective as the Ioplex that delivers the sustained iodine over days because of what the antimicrobial preservatives are. And frankly, iodine is the best anti-biofilm material agent we can use at this point that we have.
Got a few online questions. Does a lower concentration of iodine liquid still exceed the line into the cytotoxic level?
Sort of, yeah. Again, it depends on the concentration, how much you dilute it, but also remember the betadine has a detergent, a surfactant. And frankly, that surfactant can be very cytotoxic. Betadine works great when you have intact skin, you're getting ready to do surgery and you're disinfecting the skin. In fact, you kind of like the detergent there because it helps to solubilize the keratinized epithelial cells, helps to carry them off and get rid of the bugs that are there. That's a different situation when you have a wound that has no keratinized epithelium, and the surfactant and the soluble burst release form of iodine can cause problems. Okay, go ahead.
Okay, as a first line of defense with foot care, before I send them to the hospital and for wound care, do you have, like, are you planning on making hydrogen peroxide, like, instead of having the liquid, swabs similar to alcohol swabs, so that I could purchase it where I don't do dressings for my clients? It's an expense. I send them to where an RN can take care of it. So would there ever be something that would be more economical? Like, I know I don't know if it'd be the same as alcohol swabs or not, that I could take with me as a mobile nurse.
Okay, that's a good question, and I'll try to make a very brief answer. That same peroxide immobilized technology can be applied to gauze and can be used. The disadvantage of that is that it provides sustained release. And if you're just wiping a wound for 1 minute, you want almost in that case, you kind of want a burst release. So if you took 3% hydrogen peroxide, put a little bit, moisten the dressing, not give a whole saturated thing, especially in the first cleansing when you're trying to get rid mainly of slough and some superficial bacteria, that probably could be okay without having too much cytotoxicity.
Okay. I was thinking with regards of keeping it sterile in a package so that it would comply with Ministry of Health standards. That's why. Less chance of any bacteria getting in there, and it just looks better under legally.
Right. And so the Drygo actually is in a sterile peel pack. So
How much are they each about?
Say it again.
How much do they cost?
Yeah, approximately.
I would suggest maybe talking to the booth after.
Okay, thank you.
Yeah.
Right.
Yeah.
Yeah. But my understanding, and it's way out of my area, is that this is competitively cost driven. So you can put that technology on there in industrial levels without a huge cost. Do we have five more minutes? Yeah, go ahead.
Okay.
Go ahead, Shelley.
Do you need to rinse after using an antimicrobial antiseptic wound cleanser before applying Ioplex?
Okay, good question. This kind of comes with compatibility. What should you not combine with Ioplex? Well, it turns out that if you're cleansing the wound surface with typical wound cleansers, and you've removed the superficial slough and superficial bacteria, remember you haven't killed the bugs deeper in the wound bed, you can put Ioplex on, and in almost all cases, it will be compatible with what you used previously. And for example, and you're going to hear a presentation by Ergo and Bosch afterwards. And again, this is common knowledge that hypochlorous acid is an incredibly effective antimicrobial agent, not cytotoxic at the levels in Bosch, but it chemically reacts in about 3 to 4 minutes, and then it's gone because it's so chemically reactive. It doesn't persist. So even if you did a very effective Bosch cleansing, which is fine, and then you put Ioplex on, there's no contraindication because there's not enough residual hypochlorous acid molecules to chemically react with the iodine.
I think we've got a question over here, and then one more quick one here, and then we're
Can you tell me the difference between white iodine and regular?
Okay, so the kind of a chemical question, what's the difference between Ioplex-based iodine and something like betadine? In a technical level, the complex of the iodine that is in the Ioplex polyurethane foam is in a complex that allows it to be sustained release. It still comes off as I2, atomic iodine, that diffuses into the wound, into the biofilm, and then oxidizes to the antimicrobial form of I minus. Okay. If you take betadine and you pour it or use it in a wound, it is all I2, but it's a burst release with a detergent. So chemically, the thing that kills in both of those products is the oxidized iodine molecule. So it's not, there isn't a different chemistry there. It's the same chemistry, but it's how it's complexed and how it's released and what it is in combination with. And remember, the detergent in betadine is pretty cytotoxic for dermal and subdermal injury cells, not for superficial. It passes FDA for superficial stuff, but that's because it's not going down in.
I'm going to take one more question over here in under a minute.
Certainly. So with a lot of exudate, if you use iodine, it's useless because it just washes away the iodine. With this Ioplex, how effective is it on a highly exudative wound?
I'm going to tell you what my clinical colleagues tell me, and that is that if they put the Ioplex or a competitive iodine containing foam dressing, that the good news is you can see when the Ioplex has released all its iodine and you can take it off. The absorbent capability of the products depends on what the foam composition is versus the iodine that's complexed in there really to a large extent. So the polyurethane foam for Ioplex has a pretty large hydration capacity. And so it can be used on, I believe the instructions for use on low or moderate exuding wounds. If it is a really, really heavily exudating wound, then you need to understand why that wound is having so much exudate. Maybe it needs to have more compression, or you need to combine it with an alginate non-microbicidal dressing behind it or whatever to help absorb the fluid. But to me, the key thing is when it's done what it can do because it changes colors. And you see the dark color changing to white color, and it's time to replace it because the iodine releasing capability is exhausted in that part of the dressing.
I'm just going to mention as well that you can actually double it. So if you're putting it on a venous leg ulcer, for example, underneath compression, you can actually double it and you'll extend the life of the
Yeah, that's a good
Ioplex as
point.
well.
Yeah. So I know the hydropheric blue dressing pretty well, and I'm not pissing all over hydropheric blue. Its effectiveness on biofilms is significantly less than the iodine releasing dressings. Doesn't mean it doesn't work, doesn't mean it doesn't have patients where it'll do a good job, but you as a clinician have to make the decision about whether you need to hit it with the most effective, highest effective dressing at the start for 5 to 7 days and then step down to hydropheric blue. Hydropheric blue has a role. In fact, and I'll do it in the last 20 seconds, my wonderful colleague, Randy Walcott, who pioneered the use of DNA identification of bacteria in the biofilm-based wound care concepts, what his data over 13,000 wound samples showed is that when the patient's first clinical sample comes in, they identify the bacteria in there, they'll have a spectrum of bacteria and in proportions, a lot of anaerobes. If they start an antimicrobial effective treatment, after 7 to 10 days, if they go back and take another biopsy and do DNA analysis of the bugs, the bacterial spectrum has shifted. And now the bugs that were frequently in a minority because the other bugs were outgrowing them. Now, if those dominant bacteria are effectively killed by whatever antimicrobial treatment is being used, it creates an opportunity for these more minor species to start expanding. And so what happens is the pattern of bacteria changes. Well, blah, blah, what does that mean? In the biofilm-based wound care document, there's a statement that says it is optimal to do DNA identification of the bioburden, use the appropriate antimicrobial treatment to address what spectrum of bacteria you have there, then don't stay with it for more than about 10 days because of this shift that is frequently seen in the bioburden spectrum. And if you stay with the same darn antimicrobial treatment for 2 weeks, the data suggests that you're going to have a change in proportion of the bacteria. Now, if the bacteria are really, 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 really low at that point, okay, they're not going to probably cause a lot of impairment of healing. But if the wound drops real fast and then sort of begins to plateau, what we interpret that as meaning is that you're not killing the new bacteria that are in there effective with whatever topical or systemic treatment you're doing. You need to change so that you don't enhance the expansion of some of the other species that were there at the first in minor amounts.
So we're just a little bit over time. I know we've got lots of unanswered questions. We'll try to answer the questions online. And certainly you can come visit the Medline booth and we can try to answer your questions there as well. So I want to thank Dr. Schultz. We learned so much. Thank you.